Hello everyone, today we're going to be covering familial adenomatous polyposis. Familial adenomatous polyposis is an inherited rare genetic disease that is characterized by having hundreds to thousands of adenomas and polyps in the colon. The average age for polyps to appear is at 16, although familial polyposis starts at the beginning of puberty. If these polyps are not removed, it will inevitably turn into colorectal cancer. Symptoms of FAP can include but are not limited to bleeding of the colon and rectum, IBS, fatigue, changes in bowel movements, weight loss, polyps in the colon, stomach, small intestines, etc., desmoid tumors, colon cancer, and other cancers of the body. Abnormalities in other areas of the body include impacted teeth, bone osteomas and growths, benign pigmented retinal lesions, as well as skin lesions and tumors. Familial polyposis is not common at all, as it is an inherited disease. It's an autosomal dominant syndrome caused by a germline genetic mutation, meaning that the occurrence of FAP is 1 to 8,000 to 1 in 18,000. Those with this disease have a 100% chance of colorectal cancer. But taking those statistics into account, this disease accounts for less than 1% of total overall colorectal cancer cases. What are the mechanisms of FAP? Classic FAP is caused by a germline mutation in the APC gene. Germline mutations are genetic variants that come from the reproductive cell, and this is the only germ cell mutation that can be passed down to offspring. The function of the APC protein is to keep the growth of cells under control. It lets the cells know when it's time to stop growing, controls the amount a cell will cleave, and dictates how the cells will interact within a localized organ or tissue. The reason for the cell overgrowth and constant cell divisions and cleavages are due to the dysfunction of the APC gene that is in the WNT and beta-catenin pathway. This pathway explains why a cell can turn into a malignant polyp, later becoming cancerous. When the WNT is activated, the frizzled receptor activates as well. Upon activation, it will bind to the disheveled protein. Next, the beta-catenin destruction complex will bind to the disheveled protein. This results in the inactivation of the B-catenin destruction complex. When that destruction complex is inactivated, it means that beta-catenin levels will increase significantly in the cytoplasm, causing the levels to also increase in the nucleus. Once these levels in the nucleus get highly concentrated, transcriptional co-activators factor in the T-cell and the lymphoid enhancing factor, known as TCF and LEF. The beta-catenin will bind to these two, then bind to the DNA to change the gene expressions in a cell. This weird binding complex will find DNA in the nucleus, then bind to the promoter region. Once binded to the promoter region, it will promote gene expression. This leads to more proteins created that are involved in gene replication. There's a checkpoint called the G1S checkpoint that requires cyclin D. Cyclin D binds to CDK4 and phosphorylates a cytoplasm protein called retinoblastoma. The job of this protein is to stop cell division by blocking this checkpoint, causing S phase, DNA replicating, to not occur. When cyclin D increases due to the increase in beta-catenin, this causes the cyclin D CDK4 complex to phosphorylate the retinoblastoma at two sites. The retinoblastoma protein is inactivated, leaving the E2F and DP, partners to the retinoblastoma, to go to the nucleus. There's another binding site in the promoter region called the E2F box. This is where the E2F and DP will activate the transcription of the genes to start the DNA replication process. There are also two copies of the APC gene, Adenomatous Polyposis coli gene. With FAP, the germline mutation will cause a missing or dysfunction on at least one of the APC genes. When there is no APC protein present or working, it causes the beta-catenin destruction complex to not function or exist at all. Beta-catenin would not be destroyed and will exhibit that high increase of levels. High beta-catenin levels mean that cell replication occurs very fast, meaning excessive cell division. The increase in growths of colon epithelial cells become masses due to the constant cell division and overgrowths, which is how polyps form. The polyp is not instantly considered cancerous, as it would have to destroy the healthy tissue. Benign polyps are not cancerous, although they eventually become malignant and later cause colon cancer. To become malignant and cause cancer, the polyp will continue to grow. This means that the masses will build and grow onto each other, leading to mutations within each mass independently. One correct and exact mutation after constant mutations over time can lead to the malignant combination, causing more malignant polyps and destroy the tissue of the colon. This explains the very high risk of colon cancer in FAP patients. When someone is diagnosed with FAP, the plan is to get endoscopies and colonoscopies, preferably yearly. This means the polyps and adenomas are cut and burnt out, and then some are sent off to be biopsied. However, though, once the masses of cells and polyps begin to grow throughout the colon, out of control, It is advised to undergo a colectomy. A colectomy is the removal of the colon and large intestine. Depending on the height of the patient and how much intestine the surgeons can work with, they may be able to do a J-pouch procedure where they can use your remaining intestines as a makeshift colon. Untreated FAP patients do not live a long life. My mom's brother let FAP kill him. He was 34 years old. My mother found out at 29 that she had FAP, had stage 3 colon cancer, needed a colectomy. She was able to get the J-pouch procedure done. I have recently found out that I have inherited this disease, although I hope that I can educate others about this disease throughout my journey of going through it. Here are some pictures of my adenomas and polyps that they found in my colon and where they are actually located. These 2 slides are my works cited and my references I used throughout this project. Thank you for your time and thank you so much for listening to my presentation.